Welcome to my channel Anjan Bios. Today we are going to discuss our second chapter, sexual reproduction in flowering plant, higher secondary second year, and here chapter 2 we are going to discuss megasporogenesis. Let's go to the class. This is the structure of a gynoecium or pistil. Megasporogenesis is the process of formation of megaspore inside the ovule. This megaspore will finally form the female gametophyte or embryo sac. As you know, ovule develops as a primordia of nucellus over placenta. You see, this is the nucellus and ovule will form here. Okay, the tissue where ovules are attached is called placenta. In the hypodermal region of nucellus, here towards the micropylar end, develops a primary archesporal cell. This cell, nucleus of this cell become prominent, and this cell prominent than the others, and this cell is known as primary archesporal cell. This primary archesporal cell grows rapidly and finally divides once. Mitotically divide hoy, it is a diploid cell and it will form two cells. This lower one primary parietal cell and the upper one, this one, is called sporogenous cell. Or primary, we may call it a primary sporogenous cell. This parietal cell divides once or more and form the wall of the megasporangium. This cell mitotically divide once or more and finally it will form the wall of the megasporangium. Then the primary sporogenous cell here, this one, we take it out. This one, the primary sporogenous cell. Remember, it is a diploid cell. This diploid cell now directly functions as megaspore mother cell or MMC. You see, this one is megaspore mother cell or MMC. Very interesting. This cells directly functions as a megaspore mother cell. Now, this megaspore mother cell undergo reduction division. One important point here. If you remember mycosporogenesis, then remember what happened in mycosporogenesis. Mycospore mother cell will form from sporogenous cell. This mycospore mother cell in mycosporogenesis undergo mitosis division, forming a large number of diploid mycospore mother cell. Then each one undergo reduction division. This is the difference. In megasporogenesis, this megaspore mother cell, this is a diploid cell, without going any further mitotic division, this diploid cell will directly form as a megaspore mother cell and undergo reduction division. As a result of reduction division, we have 4 cells. These 4 cells all are haploid and these are arranged in this linear position and known as linear tetrad. Remember, from one microspore mother cell, in microsporogenesis produce 4 haploid daughter cells, and these 4 haploid cells are known as spore tetrad. But here in megasporogenesis, from this megaspore mother cell, the 4 cells are produced. These cells are known as linear tetrad. See, 1, 2, 3, 4. In the next step, out of these 4 megaspore produced from megaspore mother cell, upper 3 or lower 3 generally degenerate. I'll show upper 3. These 3 cells degenerate, and only one cell become functional. This cell become functional, and this functional one is called functional megaspore. This is known as functional megaspore. Now, the process of the development of this functional megaspore, this one, within the nucellus here, from these cells to functional megaspore, this whole process is known as megasporogenesis. It takes place within the megasporangium. Another important point here, as out of 4 megaspores, only 1 become functional, 3 degenerate, so the development is called monosporic embryo sac development. As this functional megaspore, now it will form the female gametophyte, or the embryo sac. So development as takes place from 1 megaspore, so the process is called monosporic embryo sac development. However, P. Maheshwari, one renowned embryologist, in 1950 reported that sometimes 2 megaspore developed into embryo sac, as in Allium. So in this case it is known as bisporic, and in Lilium also all 4 megaspore developed into embryo sac. So this is called tetrasporic embryo sac development. However, in about 70% of angiosperm, the common type of embryo sac development is monosporic embryo sac development. So this is known as megasporogenesis. Now let's see what happened after this megasporogenesis. In microsporogenesis, after formation of pollen grain, when the 2-layered pollen grains are formed, exin and intine, then it will stop their further development. Microspore develop further after it will landed on the stigma of the gynoecium. But here in megasporogenesis, megagametogenesis starts as soon as the functional megaspore form as a result of megasporogenesis. This is called megagametogenesis. You see, megagametogenesis is a process by which the angiosperms produce its embryo sac. This functional megaspore, this one, it is formed as a result of megasporogenesis. We take monosporic embryo sac development. This functional megaspore is the first cell of female gametophyte. Then this cell enlarges and its size and undergoes 3 pre-nuclear division. All these divisions are mitosis, that is, they are not reduction division, as they are already contains a haploid number of chromosome. Then first division, it will produce 2 nuclei, and both the nuclei shift to the opposite end. One is in the micropylar end and another Salazar end. Two nuclei shift towards opposite pole. You see, it is the first mitotic division. Then, as a result of first mitotic division, it will form a two nuclei or two nucleate stage. Then second mitosis takes place, it will form 4 cells. 2 the upper part, 2 the lower, it is a 4 nuclear or 4 nucleate stage. Then third mitosis takes place. As a result of third mitotic division, it will produce 8 nuclei, 4 in the salazar end and 4 nuclei in the micropylar end. This is a 8 nuclei or 8 nucleate stage. Remember, functional megaspore undergo 3 prenuclear mitotic division and finally produce a 8 nucleate structure. Then 1 nuclei from each pole. These 4 nuclei, 1 from each pole, they move to the center, and 1 from this pole and move to the center. Here we have 2 nuclei in this way. So this 2 nuclei is known as polar nuclei, located in the center, and they are not fused, but a common wall is formed like this. This is called secondary nucleus or definitive nucleus. You see, this is the final structure of an embryo sac. This one from here and this one from here. So this is the polar nucleus or secondary nucleus. Then the remaining 3 nuclei at the salazar end, this is known as antipodals. You see, these are known as antipodals. These antipodals, they are the vegetative cells and degenerate soon. They become functionless and supply nutrition. However, the 3 micropylar cells, they are collectively known as egg apparatus. This is to some extent equivalent to archegonium of bryophyte or pteridophyte. You see, these 3 nuclei collectively known as egg apparatus. The middle one become larger, this one larger, and is called egg or oosphere. The remaining 2, these 2 cells, they are known as synergids. This is a cooperative cells or helper cells. These synergids bears a filiform apparatus in the micropylar region. You see, this filiform apparatus actually, it is a finger-like projections of the wall into cytoplasm. This is the main function of this filiform apparatus. Remember this, it attracts and guides the pollen tube into the synergid. When we discuss about the fertilization, then I will discuss again. Remember now, this the function of filiform apparatus is to attract and guide the pollen tube towards the egg. In embryo sac, when pollen tube reaches one synergid degenerate, this one, at the time of entry of pollen tube into embryo sac, whereas the second one degenerates soon. First, one synergid, the pollen tube, enters through one synergid to the egg, and the second one shortly degenerate as the embryo sac has received the pollen discharge. That is, this pollen tube carry the male gamete, and we will discuss it later. So this whole structure, this is from functional megaspore up to this 8 nucleate embryo sac. This whole process is called megagametophyte or formation of embryo sac. You see, here we have 8 nuclei, 3 at the salazar end, 3 at the micropylar end, and 2 in the center. This is 8 nucleate stage, but it will form 7 cells. That is, though this central nuclei do not fuse together, but it will form a common cell wall. So 3 cell at the cellulose end, 1 cell in the central with 2 nuclei, and 3 cell in the micropolar end. This is the 7-celled but 8 nucleate stage of embryo sac. Thank you.